So what if I told you we could build human tissues with magnets? I know, it sounds like something straight out of science fiction, right? But it's actually at the core of a technology that could totally change how we make new medicines. Today, we're going to dive into how this amazing research is tackling one of the single biggest problems in the entire pharmaceutical world.
All right, let's start with a question that honestly keeps a lot of scientists up at night. Why do a whopping 90% of new drugs fail? Think about that, 9 out of 10. That's just a staggering failure rate. And a huge part of the problem comes down to how we test these drugs in the first place.
You see, the issue is, well, our best models for testing are living in a flat world. For decades, the cell cultures we've relied on to test new drugs have been, totally two-dimensional. Just cells grown on a flat plastic dish. And our bodies, they're anything but flat.
So let's really dig into this flat world problem. We're going to break down exactly why growing cells in a single layer on a flat piece of plastic is actually holding back some major medical breakthroughs. Okay, think of it this way. A 2D culture, it's like a flat map of the world. Yeah, it's useful, but it totally distorts reality, right? The cells are just stuck there in a single, unnatural layer. Their interactions are all wrong. But our bodies? Our bodies are like a globe. Cells grow in 3 dimensions, they form these complex, beautiful structures, they have intricate ways of talking to each other, and they get nutrients and oxygen from all different directions. The bottom line is, we need our models to be globes. Maps just aren't cutting it anymore.
So the big question is, how do you get these cells that are used to growing flat to start building a 3D globe? Well, this is where a really brilliant what-if moment in biology comes into play. What if? What if you could just make the cells magnetic?
The secret sauce here is this tiny thing called NanoShuttle PL. It's basically a nanoparticle cocktail of iron oxide, gold, and a special polymer that latches onto the outside of cells, giving them a magnetic coating. And the best part? It's ridiculously simple. You just add this stuff to your cells, wait a few hours, and boom, they're magnetic. No crazy genetic engineering, nothing complicated. You just add it to the dish.
And once your cells are magnetic, that's when the real fun begins. You basically get to be a tissue architect. Using simple magnetic fields, you can assemble these cells into real 3D structures. And researchers have come up with 3 really clever ways to do this. You can make them levitate, you can print them into patterns, and you can shape them into specific forms, like tiny rings. It's this incredibly elegant but super powerful toolkit for building human tissue from the ground up.
Okay, let's break this down, because the ideas are actually super simple, but so powerful. First up, levitation. You just put a magnet on top of the dish, and the magnetic cells float up and clump together right at the surface, totally free from touching any plastic. Pretty cool, right? Then there's bioprinting. This time the magnet goes underneath the dish. It pulls the cells down, packing them into a specific, dense shape on the bottom. And finally, you've got ring formation. You use a ring-shaped magnet, and it guides the cells into this perfect little donut shape, which is awesome for studying things like muscle tissue that needs to contract.
So, okay, we can build these cool 3D structures. But here's the real test. Are they actually more realistic than the old flat models? This is where, seeing is believing.
And really, this is the million-dollar question, isn't it? I mean, it's one thing to make a little ball of cells, but does it actually look and act like real tissue from inside the human body?
Well, the answer is a huge, resounding yes. I mean, just look at this. What you're seeing are human brain cancer cells. After just 1 or 2 days of magnetic levitation, they go from being a flat layer to this dense, spherical 3D structure. And honestly, it already looks a lot like a real tumor.
But the proof is more than just skin deep. It goes way deeper than just how it looks. The research zeroes in on this key protein called N-cadherin. Think of it like a biological superglue that holds cells together to form tissue. Now, in those old 2D cultures, this protein is just all over the place, totally disorganized. But in the 3D levitated culture, it's right where it's supposed to be, in the cell membranes, at the junctions, gluing everything together. And that pattern is almost exactly what you see in real tumors. And this comparison is just stunning. So on the left, that's a slice from a real human tumor that was grown in a mouse, what scientists call a xenograft. In the middle, that's our lab-grown magnetic ball of cells. The red stuff you see is that N-cadherin glue. Just look at how similar the patterns are. It's incredible. The 3D culture is basically recreating the molecular blueprint of a real tumor.
Okay, now look all the way to the right. That's the traditional flat 2D culture. And yeah, it's not even close. The protein is scattered everywhere. It's completely disorganized. This is a dead giveaway that the cells are not acting like they would in a real body. This right here, this is the flat world problem in a single picture.
And this level of realism isn't just for show, you know? It's not just some cool academic exercise. It actually lets us model incredibly complex things, like how a tumor invades healthy tissue, all there in a lab dish.
So to see this in action, researchers set up something called a confrontation assay. Sounds dramatic, right? What they did was grow a little ball of green glowing cancer cells, a really aggressive brain cancer, and another little ball of red glowing normal brain cells. Then, using magnets, they just pushed them right up next to each other.
And then they just watched. And over the next few days, you can see the green cancer cells start to actively break off and invade the red ball of healthy cells. This is a perfect mimic of what happens inside a patient's brain. And now we can watch it happen in real time, we can measure it, and most importantly, we can test drugs to see if we can stop it.
So this all brings us to the bigger picture, because this isn't just some clever niche technique. It really represents a fundamental shift in how we can do biomedical research.
And the research community is definitely taking notice. A big review from 2022 looked at all the studies in this area and found a clear growing trend. More and more scientists are using these magnetic methods, especially bioprinting and levitation. It's fast becoming a go-to tool in the lab.
And honestly, it's easy to see why. The advantages are huge. You get models that are way more biologically relevant. The little spheroids form faster and more consistently. The cultures themselves are just easier to handle because, they're magnetic. And a really big one, you don't need any artificial scaffolds or gels. The cells build their own support structure, just like they do inside our bodies.
So all of this brings us to one final, really big question. By finally leaving the flat world of the Petri dish behind, are we on the verge of being able to build custom human tissues on demand? Think about what that means. Better models for disease, maybe less reliance on animal testing, and, just maybe, a complete revolution in how we find new cures. From where I'm standing, the future of medicine is definitely looking a whole lot more three-dimensional.